In the previous lecture, we established a framework for thinking about marine life by classifying organisms according to where they live and how they move in the ocean. We discussed plankton that drift with currents, nekton that swim actively through the water column, and benthic organisms that live on or within the sea floor. As we begin this lecture, we shift our focus from simply identifying these organisms to understanding how energy enters marine ecosystems and ultimately supports all of this life. The key process that introduces energy into most marine ecosystems is primary production. Primary production refers to the biological creation of organic matter from inorganic carbon. In other words, it is the process through which living organisms convert carbon dioxide and simple inorganic nutrients into the organic compounds that form the building blocks of life. In the ocean, this process occurs primarily through photosynthesis, where sunlight provides the energy needed to drive these chemical reactions. A variety of marine organisms are capable of photosynthesis. Microscopic phytoplankton drifting in the water column perform a large share of this work, but they are not the only contributors. In shallow coastal environments, larger photosynthetic organisms such as macroalgae and seagrasses also carry out primary production. Together, these organisms transform sunlight and dissolve nutrients into organic material that can be consumed by other organisms. Although several groups contribute to marine primary production, phytoplankton dominate the process across the global ocean because they occupy the vast surface waters of the open sea. For that reason, much of today's lecture will focus on phytoplankton and the environmental conditions that allow them to grow. Understanding how primary production works and how nutrients control it will help us explain why some ocean regions support abundant life while others remain relatively unproductive. Primary production refers to the biological process through which living organisms convert inorganic carbon into organic matter. In practical terms, this means transforming carbon dioxide dissolved in seawater into the carbohydrates, proteins, and other organic molecules that make up living tissue. This process represents the entry point of energy into most marine ecosystems because it creates the organic material that other organisms depend on for food. In the ocean, primary production occurs primarily through photosynthesis. During photosynthesis, organisms capture energy from sunlight using specialized pigments such as chlorophyll. That energy is then used to power chemical reactions that combine carbon dioxide and water to produce organic molecules. Oxygen is released as a byproduct of this process. These reactions store solar energy in chemical bonds within organic compounds, making that energy available to other organisms that consume this material. Several groups of marine organisms are capable of performing photosynthesis and therefore contribute to primary production. The most widespread contributors are phytoplankton, microscopic photosynthetic organisms that drift within the upper layers of the ocean. Because they inhabit the sunlit surface waters across vast areas of the ocean, phytoplankton collectively account for the majority of marine primary production on Earth. However, phytoplankton are not the only photosynthetic organisms in marine environments. In shallow coastal lagoons where sunlight can reach the seafloor, larger photosynthetic organisms also contribute to primary production. These include macroalgae, often referred to as seaweeds, as well as seagrasses, which are flowering plants that grow rooted in marine sediments. These benthic producers can generate substantial organic material locally and often support highly productive coastal ecosystems. Despite these additional contributors, phytoplankton remain the dominant primary producers across the global ocean because they occupy the vast open water regions where most of the ocean's surface area exists. For that reason, most of our discussion today will focus on phytoplankton. Phytoplankton are the primary producers responsible for most photosynthesis in the ocean. These organisms are typically microscopic, often only a few micrometers in diameter, yet they collectively form one of the most important biological communities on the planet. Phytoplankton inhabit the upper layers of the ocean where sunlight penetrates the water column. This region is, of course, known as the euphotic zone, and it is the only portion of the ocean where photosynthesis can occur. Because phytoplankton rely on light, their vertical distribution is strongly constrained by the depth to which sunlight can penetrate seawater. Several major groups of phytoplankton dominate marine ecosystems. Diatoms are single-celled algae encased in intricate silica shells. Dinoflagellates are another important group that often possess flagella, allowing limited movement through the water. Cyanobacteria are photosynthetic bacteria that play a major role in productivity in nutrient-poor ocean regions. One remarkable feature of phytoplankton is their rapid rate of reproduction. Under favorable conditions, phytoplankton populations can double in a matter of hours or days. This rapid growth allows phytoplankton populations to respond quickly to changes in environmental conditions such as nutrient availability or light levels. Because phytoplankton grow quickly and are consumed rapidly by grazers, their populations are constantly turning over. This rapid cycling of biomass is one reason phytoplankton plays such an important role in ocean ecosystems and global biogeochemical cycles. Light is the primary energy source that drives photosynthesis in the ocean, and therefore it represents one of the most fundamental controls of primary production. However, sunlight does not penetrate very far into seawater. As light enters the ocean's surface, it gradually is absorbed and scattered by water molecules and suspended particles. Because of this absorption, light intensity decreases rapidly with depth. The upper region of the ocean, where enough light remains to support photosynthesis, is called the euphotic zone. In clear ocean water, this zone may extend to depths of roughly 100 to 200 meters. In coastal waters where suspended sediments or phytoplankton are abundant, the euphotic zone may be much shallower. Below the euphotic zone lies the aphotic zone, where light levels are too low to support photosynthesis. Although organisms may still inhabit these deeper waters, they must rely on organic matter produced in the sunlit surface layers. Light availability therefore sets a fundamental vertical boundary for marine primary production. Phytoplankton must remain within the illuminated portion of the water column in order to photosynthesize. Physical processes such as mixing and stratification influence how long phytoplankton remain within this lighted zone, which in turn affects productivity. Understanding the distribution of light within the ocean is therefore essential for understanding where phytoplankton can grow and where primary production can occur. While sunlight provides the energy required for photosynthesis, phytoplankton also require a variety of chemical nutrients to grow and reproduce. These nutrients supply the essential elements needed to build cellular structures, enzymes, and genetic material. In other words, sunlight may provide the energy for life, but nutrients provide the raw materials for which cells are constructed. Without an adequate supply of these materials, phytoplankton cannot produce new biomass even if light is abundant. Among the most important nutrients in marine ecosystems are nitrogen and phosphorus. Nitrogen is a key component of amino acids, which form the building blocks of proteins. Proteins are involved in nearly every cellular process, including metabolism, enzyme activity, and structural components of the cell. Nitrogen is also required for nucleic acids such as DNA and RNA, which carry genetic information and regulate cellular function. Because of these roles, nitrogen availability often directly controls how quickly phytoplankton populations can grow. Phosphorus plays an equally important biochemical role. Phosphorus is a component of molecules such as ATP, which stores and transfers energy within cells. It is also an essential part of DNA, RNA, and the phospholipids that form cellular membranes. Without sufficient phosphorus, cells cannot effectively store energy or maintain the structural integrity of their membranes. As a result, phytoplankton growth may slow or stop when phosphorus concentration becomes very low. In addition to these macronutrients, phytoplankton also requires certain trace metals in very small amounts. One of the most important of these is iron. Iron, which plays a critical role in the enzymes involved in photosynthesis and cellular metabolism, and in some regions of the ocean, extremely low iron concentrations can limit phytoplankton growth even when nitrogen and phosphorus are abundant. Another nutrient that is particularly important for certain phytoplankton groups is silicon. Silicon is required by diatoms, a major group of phytoplankton, to construct their cell walls. These cell walls are made of silica and form intricate glass-like structures known as frustules. In regions where dissolved silicon becomes depleted, the diatom populations may decline even if nitrogen and phosphorus remain available. This illustrates that different phytoplankton groups may be limited by different nutrients depending on their physiological requirements. Because nutrients are required for building cellular material, their availability often dominates the overall productivity of marine ecosystems. If nutrients are scarce, phytoplankton growth becomes limited regardless of how much sunlight is available. For this reason, biological oceanographers pay close attention to nutrient concentrations when studying marine productivity. In many parts of the ocean, the supply of nutrients ultimately determines how much organic material can be produced and therefore how much energy becomes available to support higher levels of the marine food web. As you can see from this illustration, nutrients enter surface waters through several mechanisms, including the mixing of deeper nutrient-rich waters, decomposition of organic matter, and inputs from rivers or atmospheric deposition. The balance between nutrient supply and biological consumption strongly influences the productivity of marine ecosystems. In many marine environments, Phytoplankton growth is constrained by the availability of specific nutrients. This concept is known as nutrient limitation, and it represents one of the central principles of biological oceanography. Nutrient limitation occurs when the concentration of a particular nutrient becomes so low that it restricts phytoplankton growth even if other nutrients remain abundant. In this situation, increasing the supply of the limiting nutrient would lead to increased productivity. In most marine environments, nitrogen is the nutrient that most commonly limits phytoplankton growth. Nitrogen is required for proteins and nucleic acids, making it essential for cellular metabolism and reproduction. When nitrogen concentrations decline in surface waters, phytoplankton growth slows even if sufficient light and other nutrients are present. The map shown on this slide indicates the nitrogen sufficiency index. Areas in red are above 80%, and that indicates enough nitrogen to not limit primary production. Values under 80% represent areas where, on average, nitrogen becomes a limiting factor to the amount of primary production. We can see the North Atlantic has sufficient amounts of nitrogen, whereas primary production in the Northern Pacific is limited by the amount of nitrogen. In some parts of the ocean, phosphorus may become the limiting nutrient. This situation is particularly common in certain coastal regions and semi-enclosed seas where nitrogen inputs are relatively high. The identity of the limiting nutrient can vary depending on environmental conditions, biological demand, and the supply of nutrients from deeper waters or terrestrial sources. Recognizing which nutrient is limiting helps scientists understand why productivity differs among ocean regions and how ecosystems may respond to environmental change. Nutrients in the ocean are not used only once and then lost. Instead, they are continuously recycled through biological processes that allow nutrients to remain available for repeated use by the phytoplankton. When phytoplankton are consumed by zooplankton, or when organisms die, organic matter begins to break down through microbial decomposition. Bacteria and other microorganisms play a crucial role in this process. As they break down organic compounds, they convert complex organic molecules back into the simpler inorganic nutrients such as ammonium, nitrate, and phosphate. These dissolved nutrients are then released back into the surrounding seawater, where they become available once again for uptake by phytoplankton. This recycling process allows nutrients to be used multiple times within marine ecosystems. In many ocean regions, particularly nutrient-poor open ocean environments, recycled nutrients represent a major source of the nutrients used by phytoplankton. In other words, the productivity of these ecosystems depends heavily on efficient recycling within the surface waters. However, not all nutrients are recycled in the surface ocean. Some organic materials sink into deeper waters before it is fully decomposed, effectively removing nutrients from the surface layer. This sinking process contributes to the vertical structure of nutrients within the ocean. Understanding the balance between nutrient recycling and nutrient loss is essential for understanding how marine productivity is sustained. One of the most consistent patterns observed in the ocean is the vertical distribution of nutrients within the water column. Surface waters, where phytoplankton live and photosynthesize, often contain relatively low concentrations of nutrients. In contrast, deeper waters typically contain much higher nutrient concentrations. This pattern arises from the interaction between biological activity and physical ocean processes. In the sunlit surface layer, phytoplankton continuously absorb dissolved nutrients from the surrounding water in order to grow and reproduce. As a result, nutrient concentrations in these waters become depleted over time. When organisms die or produce waste, organic material begins to sink through the water column. As this sinking material is decomposed by microbes, nutrients are released back into the surrounding water. Much of this decomposition occurs at greater depths, which leads to the accumulation of nutrients in deep water masses. The result is a vertical gradient in nutrient concentration, low nutrients near the surface and higher concentrations at depth. For phytoplankton to continue growing in surface waters, nutrients must somehow be returned from those deeper layers up to the euphotic zone. Physical ocean processes such as vertical mixing, upwelling, and turbulent circulation can transport nutrient-rich deep water back toward the surface. These processes therefore play a crucial role in sustaining marine productivity and trophic cycles. Although biological processes determine how nutrients are used within marine ecosystems, the supply of nutrients to surface waters is largely controlled by physical ocean processes. This connection between physics and biology represents one of the defining features of ocean ecosystems. One important mechanism that supplies nutrients to the surface ocean is vertical mixing. When wind or turbulence causes water masses to mix vertically, nutrient-rich deep water can be transported upwards into the euphotic zone where phytoplankton reside. This mixing replenishes nutrients that have been consumed by phytoplankton growth. Another particularly important process is upwelling, which occurs when deep water rises toward the ocean surface. Upwelled water is typically rich in nutrients because it originates from deeper regions where organic matter has decomposed and released nutrients back into the water column. When this nutrient-rich water reaches the surface, it can stimulate rapid phytoplankton growth. In contrast, strong stratification can limit nutrient supply. Stratification occurs when water masses form stable layers based on differences in temperature or salinity. When stratification is strong, mixing between deep and surface waters becomes limited, preventing nutrients from reaching the euphotic zone. These physical processes create distinct patterns of productivity across the global ocean. Regions where nutrient supply is high often support intense phytoplankton growth, while regions with limited nutrient input may remain relatively low in productivity. The combined effects of light availability, nutrient supply, and physical ocean processes produce clear patterns in marine productivity across the global ocean. Some regions support extremely high levels of phytoplankton growth, while others remain relatively low in productivity. Coastal regions often exhibit high productivity. Shallow waters allow sunlight to penetrate through much of the water column, and coastal processes frequently supply nutrients to surface waters. River inputs, tidal mixing, and upwelling across continental margins all contribute to nutrient availability in these environments. Upwelling regions represent some of the most productive areas in the world's oceans. In these regions, winds and ocean circulation patterns bring nutrient-rich deep water to the surface. The sudden availability of nutrients can trigger large phytoplankton blooms, which in turn support abundant fish populations and productive marine ecosystems. In contrast, many areas of open ocean are relatively nutrient poor. These regions are often strongly stratified, which prevents nutrient-rich deep water from mixing into the surface layer. As a result, phytoplankton growth is limited despite the presence of abundant sunlight. Understanding these patterns of productivity helps explain why certain ocean regions support dense marine life while others appear relatively sparse. These patterns also form the foundation for understanding how energy moves through marine food webs. So today we explored how primary productivity or primary production introduces energy into marine ecosystems. We saw that microscopic phytoplankton converts sunlight and dissolved nutrients into organic matter, forming the base of most marine food webs. However, this productivity depends strongly on environmental conditions, particularly the availability of light and nutrients. We also saw that physical ocean processes play a crucial role in controlling nutrient supply to the surface ocean. Mixing, upwelling, and stratification all influence whether phytoplankton have access to the nutrients required for growth. As a result, marine productivity varies widely across different ocean regions. In the next lecture, we will build directly on these ideas by examining plankton dynamics. Instead of focusing only on the environmental conditions that support phytoplankton growth, we'll explore how plankton populations change through time, including the formation of plankton blooms, grazing by zooplankton, and the biological interactions that shape plankton communities.